Hello, Wayne County Community College students. I'm Dr. Morrison. We're here on chapter 3, right? Tools of the laboratory, methods of studying microorganisms. So we're going to look at some different methods on how to look at microorganisms, how we're going to study them. First thing I'm going to do is go through these 6 I's. I need you to know these 6 I's in order, right? So I think I've written them in order, but if not, just check the book because I kind of just went by memory. So we'll see. Inoculation would be first. Of course, you have to introduce the microbial sample into the specific medium that you want to grow it in. So inoculation is the introduction of a microbial sample into a culture medium. And we're talking about it could be in a liquid medium known as broth. It could be on a solid medium or semi-solid medium known as the agar plate that's in the petri dish. Things like that. Whatever medium it is, liquid or solid. And the microbial sample would be maybe you took a swab with a Q-tip from your table and you put it in the broth, or you wiped it on the agar plate, or you took a loop after you flamed it and get all the microorganisms off of it. You put it into some broth of E. coli, and then you smeared that on a plate. That is inoculation. That is when you put something, introduce a specimen into a sample medium. All right, that's inoculation. Incubation. This is after you've inoculated your medium, whether it's a tube or a plate or whatever it is. You're going to place the inoculated medium into a specific environment, or you're going to put it under a specific condition to allow that whatever you inoculated to grow. So we'll just say it's E. coli because that's a real easy one for me to say. Escherichia coli, we shorten it and call it E. coli. Say it's an E. coli, we inoculated a petri dish. Now we're going to put this petri dish with the E. coli on it into an oven. It's going to incubate. That's what this is. And we usually have it set for 37 degrees centigrade for 2 days, 48 hours. So usually if you're a Monday class, we inoculate the petri dish on Monday. We put it in the oven, in the incubator, and we get the results, see if it grew or whatever it did. We're hoping that it grew on Wednesday. If you're a Tuesday class, we inoculate on Tuesday, incubate it on Tuesday, and then we take it out of the incubator on Thursday. So we give it 48 hours at 37 degrees centigrade. Those numbers you're going to need to know. Maybe not for this exam, but you're going to need to know. So for isolation, it's generating a single isolated colony on an agar medium using various pure culture techniques. These techniques we're going to learn later, but pretty soon, probably within the next month. But here they are: streak plate technique, pour plate technique, and spread plate technique. I know I'm introducing them now, but I need you to know that these are 3 methods or 3 techniques that are going to be used to isolate pure cultures, okay? We'll learn how to do these later, but at least you'll know that these are the 3 methods or 3 techniques. Inspection. Inspection, you're going to observe for the growth of the microbes. And what are some indications of growth? Well, of course, if you're looking at a liquid, also known as a broth medium, you will look for cloudiness or turbidity. Initially, the liquid is an amber to pretty kind of clear liquid, but then after you inoculate, meaning you put your specimen in that liquid, in that broth, over 2 days of incubation at 37 degrees centigrade for 48 hours, you can take it out of the incubator, shake it just a tad, and it will get cloudy. that is an indication of growth. Turbidity or cloudiness. We'll use those words interchangeably for this course. So cloudiness or turbidity in a liquid culture medium known as broth would be a great indication for growth. Another one, the presence of colonies on a solid culture medium. So if you inoculate a plate, petri dish with the solid agar there, you will start to see these little humps or clumps that grew on it. That would be indication of growth or colony. So here's the plate. First it's sterile, nothing on it. Of course, it just has the agar. And then 2 days later, you come back and you have these colonies on it. So a colony are clumps or mounds of microbial cells growing together on an agar surface. And so you could count these colonies, 1, 2, 3, 4, 5, 6, 7, 8. That would be 8 colonies, indicative of growth. So on inspection, I can see that, oh, I inoculated that sterile plate and something grew on it. You can see the colonies, right? Information gathering. This is to provide specific data and generate an overall profile of the microbes. In other words, you start looking at their shape, their size, their color, and you can make an identification of that later. You can start to, as you start to gather this information, it will start to add more to your profile. Then now we go to identification after going through all these steps: inoculation, incubation, isolation, inspection, and information gathering. Now we get to identification, and this is going to use various microscopic and biochemical procedures to characterize and identify the pure culture, right? I'm going to talk about pure culture a little bit later. First, we can look at the colony morphology. The morphology is the shape. So how does this colony look? Oh, it's kind of round. The shape, you can see the appearance and the shape of the colonies on the agar. That would be the morphology. Is it kind of shiny looking or is it dull? Does it look like, if it looks kind of hairy, that might mean you got some mold contamination in there. So we'll look at that cell morphology. Now you look at the cell shape when viewed under the microscope. We're not talking about the colony morphology. This is the colony morphology. Now we take a little bit of that from the colony, put it on a slide, prepare the slide properly, then look at the cell morphology. Is it a rod? Is it a cocci? Is it like a spirochete? Is it spiral? These are different shapes of the cells when viewed under the microscope. Next thing is cell configuration and arrangement under the microscope. How are they? How do they look? Are they clumped together? Are they in a string? Are they in a tetrad? Are they just two of them together, like paired? How are these cells looking under the microscope? And then D, various staining techniques are going to also be used. We're going to be talking about gram staining, simple staining, acid-fast staining, endospore staining, capsular staining, and negative staining. All of these types of staining are going to help us identify our specimen of bacteria. You can do a biochemical characteristics as well. That will be, does that specimen ferment sugar? So you could do a sugar fermentation test. Does that specimen utilize oxygen? So you can use the oxygen utilization test, the effect of temperature, pH, osmotic pressure on the specimen, and also you can look at some genetic characteristics of the specimen. So all these things will be included for identification. Different staining techniques, of course, will be discussed as we move on. The number one thing you're going to really have to know and memorize is going to be the gram stain. The gram staining will help you determine whether the bacteria is a gram positive because it will stain purple, or if it's a gram negative because it will stain red. I would say pink, but I don't want to give you too many P's. I want you to relate purple to positive. So PP, purple to positive, and we'll just say gram negatives stain red, okay? We'll come back and talk a bit, well, I'll talk a little bit right now about some simple stain. Simple stain is all the cells are dyed the same color. Like if you use methylene blue, all the cells are going to be blue. If you use a malachite green, I guess it'd all be green. But all cells dyed the same color, but you could look at the shape and the size and the arrangement of the cells. That's what will be important. Acid-fast, for acid-fast stain, the bacteria is going to appear pink, but non-acid-fast bacteria will appear blue. So we usually use an acid-fast to detect Mycobacterium. And a Mycobacterium is really, that's a genus with Mycobacterium tuberculosis. But we would use a different Mycobacterium, not one virulent like a Mycobacterium tuberculosis, but another type of Mycobacterium. So with endospores, remember the two bacteria that we talked about that create endospores? We have bacillus, and of course bacillus can cause anthrax, so Bacillus anthracis, which is a deadly spore that you can inhale in your lungs and it'll take over and you won't be able to breathe. Then there's Clostridium. Clostridium has, it can cause tetanus, or there's Clostridium tetani, Clostridium, I think, perfringens, Clostridium botulism, and then this one called Clostridium difficile, and you guys know it as C. diff. And people have told me that if you smell that, you'll never forget it. So I've never smelled it, so I don't have to forget it. But I have heard that from some students. Another thing is talking about capsular staining. For us, Klebsiella will be the bacteria that we will use to see capsules. And when you see a capsule in a bacteria or a bacteria that have capsules, capsules are very protective against phagocytosis. In other words, capsules protect that bacteria from being eaten by macrophages or like our white blood cells or something that comes to get rid of it in our immune system. So capsules are very protective against phagocytosis. That's something for you to remember. And the bacteria that we're going to be looking at for that will be Klebsiella. But there's sometimes some, like Cryptococcus is a fungus that would have a capsule, but we're not doing that. A negative staining, you can really see the morphology, the shape when you do a negative stain. And that's really cool because you use nigrosin, so you use like a black kind of dye, and you smear it on the plate over your bacteria and you let it dry. And then it looks just like a starry night. It looks like stars kind of poking through a black midnight sky. And it's really pretty, but you'll get to see if you have rods or cocci, and it's an easy way to check for morphology. Now we'll go on and talk about culture media. We talked about culture media already, you know, but it's also known as culture media, nutrient media, or medium containing nutrients for microbial growth. Different classifications. There are different classifications, and they can be based on the physical appearance of the media, the content of the media, like what's in it to make certain things grow and other things not grow. We'll talk about that in a minute. And the use or function of the media. The physical appearance of different media, of course, you already know. You have a liquid media. This liquid at room temperature. No solidifying agent will be in this liquid. And a liquid medium is referred to as a broth. And that's usually in a test tube. Okay? Solid medium, this will have a solidifying agent. And it is solid at room temperature. And usually we call that an agar. So that's usually in a petri dish, but you can have some agars in test tubes as well. Agar is preferred to gelatin because agar is non-reactive, meaning it is inert. Agar is stable. Agar is not used as food. It's not a source of nutrition for bacteria. It is not. And is not metabolized by most microbes. Agar will melt at 100 degrees centigrade and solidify at 42 degrees centigrade. It is a starch, really. It's a polysaccharide extract of algae. Agar is essential for the development of discrete, isolated colonies, like when we showed here. This will be on an agar plate. Another thing about medium, real briefly, is that there are medium that are called selective medium. This contains one or more agents that inhibit the growth of certain microbes, but not the other. In other words, it's going to be selective. You're going to decide that you want to grow gram negatives, right? So you're going to put in your medium things that will prevent gram positives from growing because you only want gram negative bacteria to grow. So for example, if I say, so Staph aureus is a gram positive organism. And on the other hand, E. coli is a gram negative. Well, I want all gram negatives to grow. So I'm going to put something in this medium so that it will prevent the gram positive Staph aureus from growing and only be selective towards the E. coli, the gram negatives, to grow. So this is called a selective medium, right? Selective mediums will hasten or quicken the isolation by suppressing the unwanted background organisms, in this case all the gram positives, and allow the desired growth, allowing the growth of desired gram negatives to grow. So it's going to be selective. You can put things in the medium to prevent some organisms from growing, but it will enhance the organism that you want to grow. So that's called selective medium. On the other hand, you have differential. And you have to know the difference between selective and differential, although some mediums can be selective and differential. But anyway, here's the definition of differential. Differential medium allows several types of microorganisms to grow, but they're designed to bring out the visible differences among these microorganisms. So you may see that these organisms, a lot of organisms can grow, but what's different about them? There'll be different colors, perhaps. Differences show up in variation in colony size, and the way the colony looks, or the color of the colony. So what is different, the differential? All of these colonies can grow. So it's not being selective, not picking and choosing which one can grow like selective does. And differential, all of them can grow, but you're going to be able to tell the difference between them based off of how their colony looks, their shape of the colony, or the color, right? So that's selective and differential. And you know, some mediums can be both. So I just want you to know that. Another thing about the different mediums, these agars that we can make, this one, let's see if I can just kind of find some space here to write these words up. So this agar Sabarans agar. This kind of agar is specific to growing fungus. So make sure you know that. When you see this agar, automatically think, oh, we're going to grow some fungus. Yes, you will. It cultivates and grows fungus. McConkey agar, or we call it MAC, M-A-C. This is an agar that will, like, it prefers to grow gram negatives, okay? As well as this one, eosin methylene blue, and we call it EMB. This also is selective for growing gram negatives, right? So these two, McConkey, MAC, and EMB, grow gram negatives. Mannitol salt, agar, or we call it MSA. MSA has a high salt content, and it has a preference for the genus Staphylococcus. So when you see MSA and you see some growth on it, especially if you see something that's a little yellow, it means that that is going to be Staphylococcus, because that's what it grows, right? Just to throw this other one here, sulfur indole motility. This is SMI, we call this the SIM. We put it in a tube. You can see the word motility. So this agar is to isolate what types of bacteria are motile, can move. Usually they'll have some flagella, or maybe it's just cilia, I don't know. But this is the one, and this also produces a gas, hydrogen sulfide, H2S, hydrogen sulfide. Those are an introduction to some different types of agar. You have blood agar, which has sheep blood in it. Just about everything grows on blood agar. And then TSA, that's also a very basic type of agar that just about all bacteria grow on too. They're not selective, to say the least. I wanted to use this word, fastidious bacteria, because you may see that, F-A-S-I-D, fastidious bacteria. This is bacteria that require special growth factors and very complex nutrients and organic substances. For example, chocolate agar, which we will not be using, but it really is just blood. Chocolate is not chocolate. They call it chocolate agar, but it has nothing to do with chocolate. Chocolate agar is a common medium that's used for growing fastidious bacteria. Because fastidious bacteria is like, it just requires special things to grow. So we just say it's fastidious. And chocolate agar is an example of an agar that is for fastidious bacteria. Lastly, let's see. When we talk about pure culture, let me just erase this, just so you can get an idea about a pure culture versus a mixed culture versus a contaminated culture, okay? I know this board is moving all over the place. All right. So let's start with pure. Pure culture is going to be only the microbes that come from the same same sample. So let's just use E. coli, for example. So here we have a broth in a tube, and you always label everything so you know what you're doing. So we label it and always underline it, but on this little tube you probably wouldn't. So E. coli is a broth, and this is liquid. And so we're going to flame our loop, and we're going to go in and get some E. coli. Then we're going to smear that on the Petri dish. And then on that Petri dish, we're going to get some colonies. And we're gonna know, oh, that's E. coli. That's pure, because we started off with the E. coli, we put it on the plate, and we incubated for 48 hours at 37 degrees centigrade, and boom, those little colonies are E. coli. So that's pure. Mixed is gonna be 2 or more easily differentiated organisms, right? So, let's do this. In this broth, let's have E. coli and Staph aureus. So we got 2 in here, and typically you should underline them. So that's 2. This is mixed, but we know what they are, so it's a mixed culture. And so then we flame our loop, put it in, and we spread our plate, inoculate our plate, incubate, and then let's just say 48 hours later, we get some E. coli, and we get some Staph aureus. That's fine, because we know that we started off with E. coli and Staph aureus, right? All right, so that's mixed. We know exactly what they are. So this is pure. Started off with E. coli, you inoculated, you got E. coli. Oops. You got mixed, you got 2 or more bacteria, and that's what you ended up with. Now, contaminated, totally different thing. Contaminated, right? So, I'm gonna start off with E. coli, at least that's what I think I got, in a broth. I'm going to inoculate the plate, right? And then we're gonna look, and I'm gonna get my E. coli. But then I'm gonna see some furry kind of stuff, and I'm like, what the heck? So I got the E. coli, that's for sure. But then I also have some furry mold contaminant. And so that mold is contaminant. I wasn't expecting that, right? So those are some examples of, I forgot to flame my loop and put it in here and spread it on here, okay. So those are some examples of different cultures. So I think for this chapter, as far as my notes are concerned, that's a pretty good overview. I could take a look at some other things that, in particular, I'm just gonna flip through that I want you to be pretty sure about. Know about mordants, M-O-R-D-A-N-T. Know how E. coli will appear if the mordant is not applied. About pure culture and mixed culture, know those cultures. Know all the 6 I's that we went over. Let's see. Motel, solidified. I think really go over your acid fast. What did I mention that? Oh, so differential stains, I forgot to mention. So when I talked about differential, I need to mention that there are 3 differential stains. I'm glad I saw this. Gram stain, which is one you'll have to memorize, the gram stain. The next one is acid fast stain and the endospore stain. So differential stains use dyes of contrasting color. So the dyes are different colors, like you could use, for gram stain, you use crystal violet and saffron, and so you use like a violet, purplish color, and then you also use a kind of a red color. And like I said, a gram stain will show you if the bacteria is a gram positive or a gram negative. So the two stains you have to use have to be a purple and a red. All right. So the use dyes of contrasting color clearly emphasize the differences between the two cell types or the cell parts, in addition to the size, shape, and arrangement. So differential stains are gram stains, acid-fast stains, and endospore stains. Those three differential stains you will need to know and know how they are different. Let's see, anything else in here? I think just reading through, going over the microscopes, like bright background on a microscope will be bright field, meaning the field is bright. Dark field microscopy or microscope will have a dark field. That microscope will be dark. Go over, like if you illuminate a species and it's pretty bright, then you will know that it is fluorescent. If little things that you can't see, generally that has the highest magnification, the highest resolution, that's going to be an electron microscope. Quite frankly, we didn't even know about viruses. We knew that something else existed besides bacteria, but what were they? We weren't quite sure. And then somehow invented the electron microscope, and voila, we could see these really small critters called viruses. So they have a very high resolution. What else? Know a little bit about chocolate agar. Know about basic dyes. I'm just going through this last part of the exam to just go over it. Make sure you know what the parts of the microscope are used for: the condenser, the objective lens, the nose piece, the ocular, the body, and I said basic dyes, right? And I think that is really about it. Know about TSA, that kind of agar, and blood agar. And I think that'll do it. I think for chapter 3 is basically you just go through this lecture that we went through. I pretty much summed up everything. Read the captions under the pictures. Look at the pictures. Try to understand them as best you can. And if you have any questions, then we'll talk about them in Zoom. All right. I think that concludes chapter 3. Chapters 1, 2, and 3 of the text will be on the first exam. I'll see you next time.